Hello everyone, Dr. Suresh here, your Games Academy faculty for Biochemistry. Welcome to our Fmge Biochemistry Rapid Revision for the session July 2023. So as a part of the session, I would like to summarize few of the questions which have been repeatedly tested in our previous Fmge exam. So to start with, first question. A person has a history of alcoholism for 20 years. He avoids alcohol for 2 days and presents with agitation, global confusion, disorientation, hallucination, fever, high BP, diaphoresis, and autonomic hyperactivity. The most common cause. So the options are like A. Delirium tremens, B. Wernicke encephalopathy, C. Korsakoff syndrome, D. Pancreatitis. So four options have been provided. So out of all these, one option you can easily vomit out, that is pancreatitis, because we don't have any other things related with the alcoholism and pancreas. So pancreatitis you can avoid. That means option D you can avoid. And we'll see the rest of the options like Korsakoff syndrome, Wernicke encephalopathy, and delirium tremens. So first we'll discuss about the key words that have been mentioned in the stem of the case. They have mentioned he avoids alcohol for 2 days. That means they are all the symptoms of alcohol withdrawing. Okay, so these are the symptoms of alcohol withdrawing. So we have already gone through the symptoms like global confusion, hallucinations, and all in vitamin B1 deficiency, which causes Wernicke encephalopathy and Korsakoff syndrome. So here, Wernicke encephalopathy and Korsakoff syndrome is because of B1 deficiency. But the difference between these two, Wernicke encephalopathy is acute thiamine deficiency, because this is acute thiamine deficiency, whereas Korsakoff syndrome is chronic thiamine deficiency. So this Korsakoff syndrome you can see exclusively in alcoholic people. Okay, and why alcohol people will get Korsakoff syndrome? Because we have already learned that the vitamin B1 absorption interfered by alcohol. Alcohol inhibits the thiamine absorption in the intestine. So as people are chronic alcoholic, their consumption of alcohol on regular basis prevents them for absorption of thiamine, and they get exposed to thiamine deficiency. But in case of Wernicke encephalopathy, the diet itself is lacking thiamine. Okay, whatever the diet they're consuming, that itself is a deficient of thiamine. So that is the reason Wernicke encephalopathy is acute one, whereas Korsakoff syndrome is chronic. Though there is a thiamine in the diet, the alcohol is interfering with the absorption of thiamine. Right. So now in these two, the symptoms, if you see, GOA, the Wernicke encephalopathy, like the symptoms are like GOA, that global confusion, ophthalmoplegia, and ataxia. Whereas in Korsakoff syndrome, again, complete memory loss will be there. Okay, and confabulations like telling lies, fabricating stories. Okay, and amnesia. Here, complete memory loss you can see. Amnesia is also there, and confabulations like telling lies. Okay, that is the difference between Wernicke encephalopathy and Korsakoff syndrome. In Wernicke encephalopathy, there is no complete loss. But here we have only one thing, global confusion. So Wernicke encephalopathy, Korsakoff syndrome, except global confusion, nothing is there. Hallucinations you never get in Wernicke encephalopathy as well as in Korsakoff syndrome. So hallucination is there, high BP is also there. So autonomic hyperactivity also is there. So that means these are the symptoms of alcohol withdrawing syndrome. That is delirium tremens. Okay, that is delirium tremens. So these are all the characteristic feature of alcohol withdrawn syndrome. As the person is in continuous on alcohol, he's a chronic alcohol for 20 years. Okay, and he joined the rehabilitation center. He started avoiding alcohol for 2 days. So all of a sudden, as he started avoiding alcohol, what happened? So I mean, like he started with the global confusion, disorientation, hallucination. So main thing here to remember, alcohol withdrawal syndrome, you have hallucinations and autonomic hyperactivity, which you cannot see in Wernicke encephalopathy and Korsakoff syndrome. Their characteristic features are different. Like global confusion is also there in Wernicke encephalopathy, ophthalmoplegia is there, and then ataxia you can see. Whereas in Korsakoff syndrome, complete memory loss is there, amnesia, and confabulations are fabricating lies and stories you can make out in Korsakoff syndrome. So, and there is no autonomic hyperactivity you can see in Korsakoff as well as Wernicke encephalopathy, and no increase in BP as well. So here what happened, I can see in delirium tremens, GHN. So here global confusion is there, hallucination is there, and nightmares also you can make out in delirium tremens. So again, autonomic hyperactivity is like increased BP, increased heart rate, and increased body temperature, tremors. These are also you can see increased BP, body temperature, tremors, and fever. You can make out. All these are some characteristic features of delirium tremens. So whenever the case is asked, so don't go with the opinion that it is a B1 deficiency or something. Okay. Yeah. And another question, a 2-month-old boy presented with ambiguous genitalia. Okay. His blood pressure was normal. The hydroxy testosterone value was low. The probable diagnosis is. So here again it is because recent times in Fmge, most of them are case based questions. Understand? So clinical curriculum as they implemented by NMC, there most of the MCQs are based on the case scenario. So if you are thorough with your clinicals, it is easy for you to solve the questions. So biochemistry is almost integrated with the medicine and other branches of the medicine. So here the question is 2-month-old boy. So old boy, male. Remember this key. So because if you read the question carefully, the answer is slide over there only in the stem of the question. So presented with ambiguous genitalia because his sex, I mean, like his reproductive organs are not developed. His blood pressure is normal. So the dihydroxy testosterone value is low. So the probable diagnosis is asking. So options they have given: 21 beta hydroxylase deficiency, 11 beta hydroxylase deficiency, 17 alpha hydroxylase deficiency, 3 beta hydroxy steroid dehydrogenase deficiency. So these are all the answers they have given. So before going to the correct one, I'll be discussing few of the key features of dihydroxy testosterone deficiency. So here the key feature, blood pressure is normal and a boy. So the correct option will be, so male, it is male pseudo hermaphroditism, male pseudo hermaphroditism is the answer. Okay, so because this is one of the key features, so because of the deficiency of the enzyme, 17 alpha hydroxylase deficiency. Why not 21 beta hydroxylase? Why not 11 beta? And why not 3 beta hydroxylase? I'll tell you here. So here in 21 beta hydroxylase, it is a salt wasting form. Okay, it is a salt wasting form. Okay, and at the same time, hyponatremia is the future. Hyponatremia, his BP was normal, so decrease in BP is also you can make out in 21, because here what they have given, BP is normal, but in 21 beta hydroxylase deficiency, it is a salt wasting form. Hyponatremia is a key feature and decrease in BP, so that's why it is a wrong option. And in 11 beta hydroxy, this is a hypertensive form. This is hypertensive. So that means hypernatremia you can make out. Okay, and 3 beta hydroxy steroid, this one here salt wasting you can make out in 3 beta hydroxy. This is more of like female hermaphroditism. Female hermaphroditism. So here certain key points you should aware regarding the congenital adrenal hyperplasia. So what are the things here? Mainly hirsutism you can make out in congenital adrenal hyperplasia, and because of what it is, because of 21 beta hydroxylase deficiency. And all congenital adrenal hyperplasia are female pseudo hermaphroditism, female pseudo hermaphroditism. So these are the key features of congenital adrenal hyperplasia. Understand? If you remember these points, it is easy for you to answer questions like this. So here it is a male pseudohermaphroditism. So it is 17 alpha hydroxylase deficiency. The correct answer will be 17 alpha hydroxylase deficiency. Key features is a boy, BP is normal. There is no salt wasting, there is no hyponatremia or hypernatremia. And this is not a female here hermaphroditism also. So it is a male pseudohermaphroditism, BP is normal. It is not a salt wasting form. So 21 beta is also salt wasting, 11 beta and 3 beta is also salt wasting forms, except 17. So another question, drug of choice in mountain sickness. So options are thiazides are there, furosemide is there, acetazolamide is there, spironolactone is there. So what will be the thing, drug of choice in mountain sickness? So when you are going to high altitude, there is a decrease in your blood oxygen levels, so leads to mountain sickness. So respiratory compensation has to be there. So to avoid that, what? So the drug, the correct answer will be acetazolamide. How it will works, I'll tell you. So acetazolamide, how it works? Acetazolamide is a drug which inhibits carbonic anhydrase. This is the enzyme. So this carbonic anhydrase, as it inhibits, it leads to increase in hydrogen ion concentration. That means acidosis. Right. So as you say, mountain sickness, what you have to remember in mountain sickness, your partial pressure of oxygen is low. Okay, it leads to hypoxia. It leads to hypoxia. So what happened, as partial pressure of oxygen is there, increase in acidosis. So what happened in that condition? Hyperventilation will be there. So as hyperventilation, it washes CO2, washes out CO2. There is no CO2. So what happened? As CO2 is not there, so respiratory alkalosis, it stimulates the respiratory center. It depresses the respiratory center. So as respiratory center is depressed, it leads to chemoreceptor mechanisms to elevate the carbonic anhydrase levels to be production. Right. So here the main thing, acetazolamide is the inhibitor of carbonic anhydrase. This is the key point you have to remember. Acetazolamide is a inhibitor of carbonic anhydrase, which used to which used to wash out CO2 and causes respiratory alkalosis and depresses the respiratory center. Another question, which of the following shifts the given curve to left? So far we have seen the oxygen dissociation curves. You can see to right side and left side. So here hypoxia, acidosis and anemia all shifts the curves to right side, except one, that is decrease in body temperature. Okay, so know this difference. Hypoxia, acidosis and anemia, all three will shift the O2 dissociation curve to right side. So all except one, decrease in body temperature. So here, before to know the mechanism of this, you should know what is P50. P50 is nothing but partial pressure of oxygen at which 50% of RBC hemoglobin saturated with O2. That means partial pressure of oxygen at which 50% of the hemoglobin saturated with O2. So the P50 for hemoglobin, that is 28 mmHg. So it is like reciprocal, you can make out. When P50 is increased, the saturation will be decreased. When saturation is high, P50 will be decreased. So P50 and affinity inversely proportional. So hypoxia at the tissue level, anemia, O2 carrying capacity is low, and increased body temperature, increased rate of local metabolism leads to increased O2 demand, and it leads to acidosis. I mean, again, acidosis if you're talking about, which avoids the acid production. So P50 increases means affinity decreases. P50 decreases means affinity increases. At tissue level, we need oxygen, so affinity has to be low. So P50 will be high at tissue level, and affinity will be low, so that oxygen dissociation or oxygen delivery is possible. Fat sugar baby is seen in, so options: kwashiorkor, marasmus, diabetes and hypothyroidism. So fat sugar baby you can make out in, it is kwashiorkor. So option is kwashiorkor. Why? Because it is a calories maintained, but quality of food is not good. Here the baby is consuming good amount of food, but she is not maintaining the good. That means balanced diet, in the sense it's all supposed to be the good amounts of carbs, proteins, lipids. But here the baby is consuming only calories, not protein diet. So in that condition, what happened? Only carbohydrate, fat is there in the diet. So because of that, what happens? It leads to hypoalbuminemia, and leads to protein energy malnutrition, and characteristics of protein energy malnutrition: edema. Okay, so here pitting edema is a characteristic feature. Subcutaneous fat, so that's why as fat is accumulated in subcutaneous tissues, the babies appear like fat sugar, as consuming more carbohydrates, so fat sugar baby. So fatty liver is also other thing, and baby will be apathetic. Abdominal distension also you can make out here. So here in marasmus, calories deficiency, so that is a wrong option. So no diet at all, calorie itself is deficient. Diabetes and hyperthyroidism, again they are not the correct option. So fat sugar baby is seen in, and it's a characteristic feature of what? Protein energy malnutrition, kwashiorkor. So remember the difference between kwashiorkor and marasmus. Kwashiorkor, the diet is okay, I mean, like, calories are maintained but not proteins. In marasmus, food itself is not maintained, baby is not consuming good or ample amounts of diet. The micronutrient that is deficient following post-ileal reaction, post-ileal resection. So when you remove the post-ileal part, which of the micronutrient absorption will be affected? So here folate has nothing to do with that, and iron is also nothing to do with that, because iron absorption, it requires copper. These three are nothing to do with the post-ileal resection. Only the thing is B12, because it is going to absorb the ileal section, so B12 will be affected. So the correct option is B12. So here a vegetarian patient present with anemia and hypersegmented neutrophil, the most probable causes. So the options are megaloblastic anemia, iron deficiency anemia, anemia of chronic disease, and sideroblastic anemia. So what might be the answer here? So here it will be megaloblastic anemia. Why? Because the correct option is megaloblastic anemia, because here when B12 is deficient, it leads to B9 deficiency as well, leads to folate trap. And what is the logic behind of this? As B9, B12 both are absent, so B9 is especially, so there is decrease in purines and pyrimidines. So we're all aware purines and pyrimidines are required for DNA making. So without that, there is no DNA making only. So nucleotides or nucleus cannot be produced in case of B9 deficiency. So large cells like excess of cytoplasm with hyper segmented nuclei. So as nuclei is ready to divide, but because of the absence of DNA they cannot. So they started what, like especially neutrophils have more than 6 lobes. They get the nucleus occupies the excessive space of the cytoplasm. So that is the reason we have got multi-lobes in the neutrophils. So it is a clear-cut case of megaloblastic anemia. So it is not a case of iron deficiency anemia or sideroblastic anemia because of again excessive of iron overload, and anemia of chronic disease where the iron absorption will be deficient. So here another repetitively tested question in competitive exams, not only in FMG, in NEET PG also. A person presents with diarrhea, dementia, and dermatitis. So you see here three characteristic features you can make out: diarrhea, dementia, dermatitis. What is the probable cause? So diarrhea, dementia, dermatitis, these three are the characteristic features of pellagra. 3 Ds. Remember, 3 Ds leads to fourth D. So 3 Ds are diarrhea, dementia, and dermatitis, which leads to fourth D, that is death. Right. Cystinuria is because of the defect in the transport mechanism at the kidney renal, renal tubules. So it is like COLA, cysteine, ornithine, lysine, arginine. All basic amino acids will be excreting or following the cysteine. Cola urine. Alkaptonuria because of the presence of alkaptin bodies in the urine, such as homogenistic acid deficiency. More amount of homogenistic acid will be excreted in the urine. Porphyria, again it is a heme synthesis defect. So the correct option is pellagra. And pellagra, what is that? Pellagra is a characteristic feature or deficiency of which vitamin? That is B3 niacin. Okay, B3 niacin. Menkes disease is caused by which of the thing? Menkes disease, options like copper deficiency, copper toxicity, zinc deficiency, fluorine excess. So these are the two things you have to avoid. Zinc is nothing to do with the Menkes disease, zinc and fluorine. Copper toxicity is like copper overload. This is also not a correct option. Copper deficiency, yes, copper deficiency is why, because copper is there so in the cell, it which cannot be transported out of the cell because of the gene deficiency, that is ATP7A gene, which plays major role for copper absorption at intestine. So, and also which plays major role in incorporation of copper in the enzymes. Okay, so lysyl oxidase is a copper requiring enzyme. Lysyl oxidase is collagen maturing enzyme. So this copper absence makes lysyl oxidase inefficient, and collagen synthesis or collagen maturation will be hampered. So no collagen. So that's why no hair texture. No hair structure texture. So repeated bone fractures as there is no collagen in the bone maturation. So copper toxicity is Wilson's disease. And zinc deficiency is acrodermatitis enteropathica. Fluoride leads to what to say, excessive deposition of like dental fluorosis, and also like bone fluorosis also you can make out. Cholesterol is synthesized from, so again the basic question has been asked repeatedly. Cholesterol is synthesized from options acetyl CoA, bile acid, phenylalanine, heme. So here heme is synthesized from succinyl CoA plus glycine. Phenylalanine, it is a essential amino acid So it's supposed to be in the diet. It cannot be synthesized in the body. So bile acids, they are all coming from cholesterol. So cholesterol itself is the source for bile acids. Then how come cholesterol will be synthesized from bile acid? So all these three options are wrong. So except acetyl CoA. So here acetyl CoA combines with another acetyl CoA. Like that, three acetyl CoAs combine to form Hmg CoA. And finally they form 27 carbon cholesterol. Okay, so like this. And acetyl CoA is not only for cholesterol, you can make out ketone bodies also, and it can be oxidized in the TCA cycle also. So that is the reason when you are having excess of glucose in the diet, this glucose, I'll show you the fate of glucose. Turn off the charger. Glucose gives pyruvate, and pyruvate gives acetyl CoA. So suppose 100 molecules of glucose are there, so it will give 100 acetyl CoA. So this acetyl CoA has to enter into TCA cycle, right? Here when it has to go to the TCA cycle, okay, this 100 acetyl CoA is supposed to enter into TCA cycle, means 100 oxaloacetate supposed to be there, but here we have only 50 oxaloacetate. So 50 acetyl CoA will be entering into TCA cycle to form 50 citric acid molecules, and 50 of acetyl CoA diverted for cholesterol synthesis, ketone body formation based on the body demand. Because the intermediates of TCA cycle are limited, whatever the acetyl CoA is coming, they cannot be entering into TCA cycle. When you are having a sedentary lifestyle, you don't have much of the energy to be produced. So in that condition, most of the acetyl CoA will be diverted. Instead of oxidizing, they'll be diverted for cholesterol synthesis, and they'll be storing, and also lipid synthesis. Another question: what happens after a meal? What happens after a meal? So here the options. So what happens after a meal when you consume a diet? I mean, like, the diet supposed to be, the food is supposed to be digested and absorbed. It should be appear in the blood, right? So after the meal, so options will see increase in insulin and decrease in glucagon, increase in glucagon and increase in insulin, no changes in both, decrease in insulin and glucagon. So our being Indian, I mean, like, our diet is more of like carbohydrate diet. So the moment food enters into the intestine, absorption, okay, the glucose molecules enter into the blood, portal circulation to systemic circulation. Pancreas will sense, there are GLUT2 receptors in the pancreas which sense the raise in the blood glucose level, and they start releasing the insulin, right? When there is a glucose from outside, why you have to produce glucose from inside? So the option will be yes, immediately there is a raise in the blood glucose level after a meal, there is increased insulin secretion from the pancreas. Obviously there is no need of glucagon for inside glucose production, so it has to decrease, right? So increase in insulin, decrease in glucagon. Which of the following activities are low in riboflavin deficiency? Which of the following activities are low in riboflavin deficiency? Riboflavin, what is that? Riboflavin is B2, water soluble vitamin. Okay. So I'll give you one of the best thing to what to say. So here B1, B2, and B6, okay, and B12. So B1 is thiamine, TPP is a coenzyme form, B2 is riboflavin, like FADH2 you can make out, B6 is pyridoxal, PLP is this one, B12 is cyanocobalamin, so hydroxy methyl cobalamin or methylcobalamin is a coenzyme forms. So these coenzyme forms required for some of the enzyme activities. So suppose transketolase, if you see, so transketolase enzyme activity dependent on TPP, thymine pyrophosphate. So that's why in case of anemia, like what to say, they will check these TPP levels. In glycogen phosphorylase, you can make out like B6, PLP is required, and propenyl CoA carboxylase, carboxylation reactions B7. Any carboxylation reaction in our body requires B7 as coenzyme form. Okay, and glutathione reductase, there is a selenium, what to say, B2, sorry, selenium, which requires glutathione peroxidase. Know the difference. Glutathione reductase is different, glutathione peroxidase is different. Glutathione reductase, okay, dependent on B2. So the B2 deficiency will be analyzed by estimating glutathione reductase. Same way B1 deficiency will be analyzed by estimating transketolase activity. Glycogen phosphorylase activity will be measured by B6 PLP activity, and propenyl CoA carboxylase and B7. So like this, you have to remember some specific. Transketolase means B1, carboxylation reactions, propenyl carboxylase B7, glycogen phosphorylase B6 PLP, and glutathione reductase B2, riboflavin. Which of the following activities are low in riboflavin deficiency? Which of the following activities are low in riboflavin deficiency? Again, we just now discussed. Sorry, it is repeated. So, which of the following enzyme is defective in Mann-Greaves disease? So, first we will see what are the glycogen storage disorders. If you make a tabular column, type 1 and disease and enzyme deficiency. So, first one, type 1, Mann-Greaves disease, enzyme defect. Here that is Mann-Greaves disease because of the deficiency of the enzyme glucose 6 phosphatase. Okay, the enzyme is glucose 6 phosphatase. Hypoglycemia is a prominent feature. Okay, hepatomegaly again one of the prominent feature. Remember, hepatomegaly again one of the prominent feature of Mann-Greaves disease. Second one is Pompe's disease because of the enzyme deficiency acid maltase. Because if not the direct question, case based questions will be asked based on the glycogen storage disorders. Acid maltase deficiency, otherwise alpha 1,4 glycosidase enzyme deficiency. Here the characteristic feature of this disease are floppy baby, frog like posture. Okay, death also may happen in case of Pompe's disease. And Coris 3 is Coris disease. So C followed by D. So D branching enzyme deficiency. Fourth one, Anderson disease, A followed by B. So branching enzyme. Fifth one, McArdle disease, which because of the muscle glycogen phosphorylase enzyme deficiency, M to M. Exercise intolerance, one of the key feature. Sixth one is Hirsch disease because of hepatic phosphorylase enzyme deficiency. Seventh one is Torres because of the deficiency of enzyme phosphofructokinase 1. So these are the key glycogen storage disorders with enzyme deficiency. So you have to by heart, you have to have these enzymes on tip of your tongue. Mann-Greaves disease, glucose 6 phosphatase deficiency. Pompe's disease, acid maltase or alpha 1,4 glycosidase activity. Coris disease because of deficiency of debranching enzyme. Anderson disease because of the branching enzyme. McArdle disease because of the muscle glycogen phosphorylase deficiency. Hirsch disease because of hepatic glycogen phosphorylase deficiency. Torres is because of phosphofructokinase 1. So the question is, which of the following enzyme is defective? That is glucose 6-phosphatase, where glucose 6-phosphate cannot be converted to free glucose. As free glucose is not there, it leads to condition hypoglycemia. Glucose 6-phosphatase is absent in muscles. If it is absent in the liver, it leads to a condition Mann-Greaves disease. Normally, glucose 6-phosphatase is not there in the muscles because glycogen is there muscles as well as in liver. So when you want to break down the or you want to get glucose from glycogen, you have to have glucose 6-phosphatase in the pocket. So muscles don't have, liver has. If liver is also having the deficiency of this glucose 6 phosphate, you have the disease, Van Gog's disease. A 8-month-old infant presents with hypoglycemia. On examination, hepatomegaly was observed. Blood investigations revealed lactic acidosis, ketosis, and xanthomas on the buttocks. What is the most probable enzyme deficiency? So this is a recently tested question in 2021. So what happened here? Hypoglycemia is there. Examination, hepatomegaly, you can make out. Blood investigation revealed lactic acidosis, ketosis, and xanthomas. So here the enzyme deficiency will be glycogen synthesis. So how come glycogen synthesis? As glycogen is not formed, okay, as glycogen is not formed, most of the glycogen, so here no hypoglycemia, here they have not mentioned any hypoglycemia. Okay, a 8-month-old infant presents with hypoglycemia. On examination, hepatomegaly. Yes, this is a clear-cut indication. You remember hypoglycemia and hepatomegaly. Just now we discussed Van Gog's. This is a clear-cut case of Van Gog's disease, because hypoglycemia is one of the prominent feature of Van Gog's disease and hepatomegaly. So blood investigations reveal lactic acidosis. Yes, glucose 6-phosphate pyruvate will be converting pyruvate to lactate, because glucose 6-phosphate will be entering into the repeated glycolysis. So feedback inhibition, which will not allow, which will inhibit pyruvate dehydrogenase. So this pyruvate will be converted into lactic acid, leads to lactic acidosis. Ketosis is also there and xanthomas is also there. So first we'll see what are the things. So the correct answer will be glucose 6 phosphatase. It's a clear-cut case of Van Gog's disease because of two key features. One is hypoglycemia, other one is hepatomegaly, and these are all ketosis. Xanthomas are the characteristic features of Van Gog's disease. So here, as glucose 6 phosphatase is absent, so glucose 6 phosphate cannot be converted to free glucose. Remember, it cannot be converted to free glucose. So that's why they take alternative pathway. So this glucose 6 phosphate entering into glycolysis, and this will be forming pyruvate, and this pyruvate will be forming lactate, leads to lactic acidosis, and it also leads to acetyl CoA, and this acetyl CoA which will be entering into fatty acid synthesis. So that's why hypertriglyceridemia, xanthomas, okay, so hypercholesterolemia, chubby cheeks, okay, doll like futures, apple cheeks, okay, and you can say triglyceride accumulation leads to THG accumulation leads to xanthomas, and acetyl CoA again leads to ketone body formation, as there is a prominent feature of hypoglycemia. So ketosis is also again prominent feature. So the enzyme deficiency is glucose 6 phosphatase. It's a clear-cut case of Van Gog's disease. So again, this is a question. So the targeting topics for Fmge: electron transport chain. Next is glycogen storage disorders. Next is image based questions, vitamins. So if you pay attention to three of these topics, you will be like damn sure about getting 3 to 4 questions from these. One is electron transport chain, other one is glycogen storage disorders, other one is vitamins. Maximum 4 marks are in your pocket. So when you are thorough with these 3 topics, Mal bio 1 or 2 you can expect, but repeatedly they are targeting these topics: glycogen storage disorders. Next one is electron transport chain. So complex 4 is inhibited by, so rotenone is a complex 1 inhibitor, okay, Pearson and rotenone. Malonate is a complex 2 inhibitor. British anti-leukocyte agent is a complex 3 inhibitor, and cyanide is a complex 4 inhibitor. Okay, along with cyanide, hydrogen sulfide is also there, carbon monoxide is also there. So option cyanide they have given, obviously. So rotenone, pyazidine, okay, are the complex 1 inhibitors. Malonate is inhibitor of succinate dehydrogenase complex 2. British anti-leukocyte agent and anti-myosin are the inhibitors of complex 3, right? So again, sometimes they will ask uncouplers regarding question. So biological, like physiological uncouplers and chemical uncouplers. So 2,4 dinitrophenol. This is a chemical uncoupler. Biological uncouplers is like thermogenin, thyroxine, fever, are the physiological uncouplers. So what do you mean by uncoupler? Uncoupler is nothing but oxidative phosphorylation. Passage of electrons coupled with phosphorylation of ADP to form ATP. Here uncouplers, what they do, they will allow the passage of electrons. Water molecules, I mean like hydrogen radiation nucleus will be donated to oxygen to form water molecule, but they will not create proton gradient or proton pump, which will not allow hydrogens which are escape to interstitial membrane space in the mitochondria to enter again into the uh, crystal side of the mitochondria. So they block the complex 5. Uncouplers are the one that is ATP ADP synthesis, F0 F1 complex or complex 5 of electron transport chain. So all the properties of genetic code, all the following are properties except of genetic code. Degeneracy, yes. Ambiguous, not punctuated or non-overlapping. Yes, degeneracy, you can say number of codons, how many you have? 64 codons we have. So out of this 64, 3 are stop codons, one is start codon. So what are the 3 stop codons we have? UAG, UAA, UGA are the this one. And so one start codon, that is UA start codon. So 20 amino acids are there. So each amino acid may code by more than one codon. So that is degeneracy. So exception is methionine and tryptophan, which are coded by only one codon. Okay, and ambiguous, no. So this is a exception. So unambiguous, single amino acid. So here not more than one amino acid can be coded by single codon. Okay, so they are not punctuated. Yes, they are non-overlapping. Okay, genetic or non-ambiguous, not ambiguous. So the optically inactive amino acid. So the property of optical activity depend on the number of chiral carbons or asymmetric carbons. So glycine, if you see the structure of glycine. So what do you mean by chiral carbon? Chiral carbon is nothing but the 4 valencies of carbon are supposed to be satisfied by different groups, right? So here glycine is having 2 similar groups. So it doesn't have any chiral carbon, absence of chiral carbon in glycine. So that makes glycine optically inactive. The amino acid which acts as a physiological buffer. So here glycine, it's simple amino acid. Tryptophan is an aromatic amino acid. Tyrosine, again it is a aromatic amino acid. Histidine is the one which acts as a physiological buffer because it is a basic amino acid which has extra amino group which can reacts with the changes in the, it can accept the hydrogen, it can donate the hydrogen. A 40-year-old man, again case based question, this one asked in June 2020. A 40-year-old man presented with sclera of eyes and pinnae have black pigmentation. The color of the urine turns dark on standing. What is the probable cause? So here sclera of eyes and pinna have black pigmentation. Okay, and urine turns dark. So dark urine, otherwise known as dark urine disease. So this is a clear-cut case of alkaptonuria because of the enzyme deficiency, homogeneous state oxidase. So inborn errors, another favorite chapter or topic for FMG exam you can make out, that is inborn errors of protein metabolism, especially phenylalanine inborn errors, and then other like what to say, tryptophan metabolism. Okay, so phenylketonuria, it is a mousy odor. It doesn't have any characteristic features of like dark urine disease something. High levels of phenylalanine will be excreted, otherwise it's not as such directly phenylalanine in phenylketonuria. The other alternative metabolic products of phenylketonuria, that has phenyl pyruvate, phenyl lactate or phenyl acetate. Because of the presence of phenyl acetate in the urine, the person who is having phenylketonuria, his body odor or urine smells like mousy odor. Okay. In alkaptonuria, there is no as such what to say odor symptoms. They have visual symptoms like what to say, because it is a strong reducing substance. Homogenistic acid is a strong reducing substance. Because of the absence of homogenistic oxidase, these homogenistate start accumulating in the soft tissues in the body, like uh, sclera of eyes and pinnae, and start blackening the cheeks as well. But and also, if you see the um, characteristic features of urine, when you allow the presence of this homogenistic acid in the urine, it reacts with the oxygen in the air and it converts urine into dark color. So here maple syrup urine disease, because of the branched chain amino acid dehydrogenase complex deficiency. Hartman's disease is absorption of tryptophan. Absorption of tryptophan is defected here. Absorption of tryptophan is defected here. So the correct option is alkaptonuria. So this is a clear-cut case of alkaptonuria. You see sclera of eye has got dark in color. So the enzyme deficiency is homogeneous oxidase, homogenistic acid. So the metabolic product will be benzoquinone acetate, melanin, ochronosis, pigmentation of pinnae, nose, thenar, and hypothenar emensis. So these are the characteristic features of alkaptonuria. So these are the previous year questions that have been repeatedly tested. So now I would like to go with the important one line or last minute revision for FMGE. So these are I'm trying to cover like impact topics which has a high chances of testing in the exam. So first we'll start with the metabolism aspect. Okay. So carbohydrate metabolism. So first, any metabolism aspect, if you're talking about, you should aware of the enzymes and the location, the pathway where it is taking place. Okay. So glycolysis, carbohydrate metabolism first we'll discuss, and the rate limiting enzyme also. So glycolysis, otherwise known as Embden-Meyerhof pathway. The location of the pathway is cytosol or cytoplasm. The key rate limiting enzyme for this is phosphofructokinase. Phosphofructokinase 1. Okay. And Krebs cycle, TCS cycle, or citric acid cycle, which takes place in mitochondrial matrix. Okay. The key enzyme or rate limiting enzyme, that is isocitrate dehydrogenase. Isocitrate dehydrogenase. Gluconeogenesis, which eventually takes place in liver and kidney, and subcellular organelles, cytosol and mitochondria. The rate limiting enzyme is fructose 616 bisphosphatase. Remember, for glycolysis it is phosphofructokinase 1, whereas for gluconeogenesis the key enzyme or rate limiting enzyme is fructose 1,6 bisphosphatase. It not the glucose 6 phosphatase or not the pyruvate carboxylase. Remember. Okay, glycogenesis, which exclusively takes place in liver and skeletal muscle, cytosol. Glycogen synthase is a rate limiting enzyme, and glycogenolysis, liver and skeletal muscle, cytosol. The enzyme is glycogen phosphorylase. For the rate limiting enzyme for glycogen synthesis is glycogen synthase. The rate limiting enzyme for breakdown of glycogenolysis or glycogen, that is glycogen phosphorylase. HMP shunt or pentose phosphate pathway, which takes place in cytosol. The key enzyme or rate limiting enzyme, that is glucose 6 phosphate dehydrogenase. Okay, so these are the key pathways of carbohydrate metabolism and rate limiting enzyme. Once again, I am repeating. The rate limiting enzyme for glycolysis, that is phosphofructokinase 1. The rate limiting enzyme for TCA cycle, isocitrate dehydrogenase. The rate limiting enzyme for gluconeogenesis, that is fructose 1,6 bisphosphatase. The rate limiting enzyme for glycogen synthesis, that is glycogen synthase. The rate limiting enzyme for glycogenolysis, that is glycogen phosphorylase. And for HMP shunt, that is glucose 6 phosphate dehydrogenase. And for protein metabolism, here urea cycle or ornithine cycle, which is taking place in cytosol or mitochondria. Two reactions are taking place in mitochondria. Rest of the reactions are taking place in mitochondria. So the key enzyme or rate limiting enzyme for urea cycle, that is carbamyl phosphate synthase 1. Okay, there is another enzyme, carbamyl phosphate synthase 2, that will be coming in pyrimidine synthesis. Okay, the difference is carbamyl phosphate synthase 1 present in mitochondria, whereas CPS2 is present in cytoplasm. So that is a key difference, CPS1 and CPS2. CPS1 is for urea cycle, CPS2 is for pyrimidine synthesis. Okay, and lipid metabolism you are talking about, beta oxidation, again it takes place in mitochondria. Okay, beta oxidation is taking place in mitochondria. What is the main key enzyme? Carnitine acyltransferase 1 is the rate limiting enzyme here. Ketogenesis again takes place in mitochondria. The key enzyme is Hmg CoA synthase. Okay, fatty acid synthesis, cytosol, cholesterol synthesis. So here, except beta oxidation and ketogenesis, fatty acid synthesis, cholesterol synthesis takes place in cytoplasm. For fatty acid synthesis, the rate limiting enzyme is acetyl CoA carboxylase. Cholesterol synthesis, Hmg CoA reductase. Okay, and for purine synthesis, PRPP glutamyl amidotransferase, phosphoribosyl pyrophosphate glutamyl amidotransferase is a rate limiting enzyme. So carbohydrate, as I mentioned, so key enzymes you have to remember. Carbohydrate, type 1 Wang-Riggs disease, glucose 6-phosphatase is an enzyme, as well as severe hypoglycemia. The symptoms, let us see. Severe hypoglycemia, hepatomegaly, renomegaly, lactic acidosis, hyperlipidemia, ketosis, hyperuricemia. Why hyperuricemia? Why? Because glucose 6-phosphate is a starting substrate. So here lactic acidosis, hyperlipidemia, ketosis we have already learned while explaining the questions, previous year questions. Okay, and why the hyperuricemia? Why there is a relation between Wang-Riggs disease and gout? Because if you see the gout, glucose 6-phosphate cannot be converted into free glucose in Wang-Riggs disease. So most of the glucose 6-phosphate will be entering into the glycolysis, and some of the glucose 6-phosphate will be diverted to HMP shunt, which is starting substrate for HMP shunt. Another minor oxidative pathway for glucose. So what is the use of this? This is useful for nucleic acid formation. How? It provides pentoses. Glucose 6-phosphate entering into HMP shunt provides pentoses. Pentoses are useful for purines and pyridine formation. When these purines are synthesized in excess, they has to undergo degradation. So because of the high degradation of purines, they form uric acid. High levels of uric acid leads to gout. So that's why Wang-Riggs disease indirectly related to gout. And Pompe's disease, because of the deficiency acid maltase and lysosomal glucosidase enzyme deficiency. Cardiomyopathy, hypotonia, systemic findings leads to early death by 2 years. Type 3 Coris disease, debranching enzyme, similar to type 1 but milder symptoms. Normal blood lactate levels you can make out, not as lactic acidosis. Lactic acidosis you can make out in Wang-Riggs but not in Coris disease. Anderson disease, branching enzyme deficiency, similar to hepatosplenomegaly, failure to thrive in infancy, progressive cirrhosis, death usually before 5th year in Anderson disease. McArdle's, muscle glycogen phosphorylase, painful muscle cramps with strenuous exercise and muscle glycogen abnormally high. Type 6 hepatic glycogen phosphorase deficiency you can see in Hirsch disease. Hepatomegaly is a prominent feature. Essential fructosuria because of fructokinase deficiency. Fructose appears in blood and urine. Fructose intolerance because of the aldolase B. Aldolase A you can see in glycolysis, where fructose 1,6-bisphosphate will be splitted into dihydroxyacetone phosphate and glycerol 3-phosphate. Right. So aldolase B you can see in fructose metabolism. So where it cannot be converted into another form of fructose, so it leads to jaundice, vomiting, seizures, lethargy, and hepatomegaly. So galactosemia because of deficiency of the enzyme galactose 1-phosphatase uridyl transferase. Characteristic features are jaundice, vomiting, seizures, lethargy, hepatomegaly, oil drop cataract, and mental retardation. Remember, oil drop cataract is a characteristic feature of galactosemia. Sunflower cataract is also there, you can make out. So that is totally different from this oil drop cataract. And phenylketonuria, if you are talking about protein metabolism disorders, phenylketonuria, just now we discussed. Phenylalanine hydroxylase is the enzyme deficiency. Hypopigmented skin, blondie hairs, Hyperpigmented because phenylalanine is required, which forms tyrosine. Tyrosine is the main thing to synthesize melanin. As melanin is not there, hyperpigmented skin you can make out blondie hairs, intellectual disability, what to say, microcephalae, seizures, and mousy or musty odor. You can make out the characteristic feature. Alkaptonuria, black urine disease. Homogeneous state oxidase enzyme deficiency, urine turns on prolonged exposure to air, ochronosis, bluish black connective tissues, ear cartilage and sclerae of eye, maple syrup because of the deficiency of the enzyme, branch chain keto acid dehydrogenase or decarboxylase deficiency, vomiting, poor feeding, urine smells like maple syrup or burnt sugar. And lipid storage disorders, Niemann-Pick disease because of the deficiency of the enzyme sphingomyelinase, Gaucher's disease because of the deficiency of the enzyme beta glucocerebrosidase, Krabbe's disease because of deficiency of the enzyme beta galactocerebrosidase. Gluco is for Gaucher's, galactose is for Krabbe's. Fabry is because of alpha galactocerebrosidase, Tay-Sachs is hexosaminidase A, and Sandhoff's is hexosaminidase A and B. Lichtenstein syndrome, nucleotide deficiency, HGPRT. What is HGPRT? Hypoxanthine guanine phosphoribosyl transferase. Hypoxanthine guanine phosphoribosyl transferase is the uh, deficiency which leads to hyperuricemia, gout, intellectual disability, self-mutilation, red orange crystal in urine. So if you want to go much about Lichtenstein syndrome, I already started some YouTube promotional videos for Fmge case-based questions. If you are following it, you can understand better the mechanism behind of this. So gout again because of hyperuricemia, needle-shaped monosodium urate crystals in joints leads to gouty arthritis. And important products that are coming from amino acids and specialized products. So tyrosine is useful. Phenylalanine gives tyrosine. Tyrosine is useful in making melanin, T3, T4 and catecholamines. And tryptophan is required for the hormones melatonin, serotonin, vitamin B3. Yes, 60 mg of tryptophan is required to synthesize 1 mg of niacin. Remember. Histidine is required for histamine, anti-allergic substance. Arginine is for nitric oxide and creatine formation. Yes, creatine is made over by 3 amino acids: glycine, arginine, and methionine. Remember this composition also. Okay, creatinine requires 3 amino acids. One is glycine, other one is arginine, next is methionine. So again, the important products that are synthesizing from glycine, that is heme. Yes, along with succinyl CoA, glycine involves in making of heme. Purine ring, whole molecule of glycine is incorporated in purine ring. Glutathione, it is a tripeptide, gamma glutamyl cysteine glycine, which is a membrane antioxidant. Creatine, just now I discussed, along with the methionine arginine, glycine involves in making creatine. So inhibitors, electron transport chain, So fluorides, so sodium fluoride inhibits enolase enzyme, which is a enzyme of glycolysis. Barbiturates like phenobarbital and rotenone, complex 1 inhibitors. Malonate is a complex 2 inhibitor. Antimycin or British anti-lewisite is a complex 3. Carbon monoxide, cyanide and hydrogen sulfate are the inhibitors of complex 4. And oligomycin, complex 5. Okay, oligomycin is a uncoupler which inhibits phosphorylation of ADP to ATP. And regarding the classification of amino acids, there are 20 amino acids. Out of the 20, 8 are essential, 2 are semi-essential, 10 are non-essential, and 2 recent additives, selenocysteine and 21st amino acid selenocysteine, and pyrrolysine is again one more amino acid, 22nd. So aliphatic amino acids like straight chain, which are in having glycine, alanine, valine, leucine, isoleucine are the aliphatic amino acids. Hydroxyl group containing amino acids, serine and threonine. Okay, if you see the basic structure of amino acid, so wherever R is there, so they will change from here. Okay, if R is like hydroxyl group, that is hydroxyl group. If it is having sulfur group, sulfur containing. If it is having COOH, it is acidic amino acid. If it is having amino group, basic amino acids. If it is having aromatic ring, then aromatic amino acid. So this R, green color will not change. Only R will be changing based on the functional group. This R is nothing but functional group. So functional group classification. So sulfur containing amino acid cysteine and methionine. Remember, in cysteine and methionine, cysteine is having a free sulfur group, whereas methionine the sulfur group is locked. So there is no free sulfur available in methionine. The free sulfur available only in cysteine. Acidic amino acids, if you're talking about aspartic acid, glutamic acid, aspargine, glutamine. Basic amino acids, lysine, arginine and histidine. Aromatic amino acids, tyrosine, tryptophan, phenylalanine. Amino acid is proline. Remember, amino acid. The difference is amino group is there in case of amino acids. In amino acid, amino group is in linkage with the carboxylic group. So that's why it is combined with the amino group is combined with the carboxylic group. So it is amino acid. Remember, it is a amino acid. So only one amino acid is there. Okay, proline. So I'll tell you the difference also. Alpha amino acids and amino acid. So this is also important. Alpha amino acids, when they treated with ninhydrin test, they will give romance purple. That indicates presence of alpha amino acids. Presence of amino acids, when they treated with ninhydrin test, they will give yellow color. So yellow color, I have given you test sample. I want you to find out whether it is amino acid or amino acid. So I asked you to perform ninhydrin test to find out which type of molecule, amino acid or amino acid. So when you add ninhydrin reagent to the sample, if it turns to romance purple, that is alpha amino acid. Otherwise, if it gives you yellow color, then it is a amino acid, that is presence of proline. So out of the 22, okay, so I can say essential amino acids 8. Okay, what are they? Tryptophan, valine, threonine, okay, isoleucine, leucine, lysine, phenylalanine, methionine, and 2 are semi-essential. Semi-essential, arginine and histidine are semi-essential. And ketogenic amino acids are leucine and lysine. Lysine is purely ketogenic. Remember, lysine is purely ketogenic. Glucogenic and ketogenic amino acid, tyrosine, tryptophan, phenylalanine, isoleucine. Glucogenic amino acid is rest of all are. The 21st amino acid is selenocysteine. Remember, 22nd is pyrrolysine. Essential fatty acid, linoleic acid, linolenic and arachidonic. When you are having linolenic acid, you can synthesize arachidonic acid and linoleic acid from both. Okay. And now to talk about minerals. Okay. So micro minerals and macro minerals we have already learned. What are macro and micro? Macro minerals, if the requirement is more than 100 mg per day, okay, 100 micrograms per day requirement, they are macro minerals. If the requirement is less than 100 micrograms per day, then they are known as micro minerals. Okay, so first we have to see most of the cofactor functions of the enzymes depend on these micro minerals. So first we'll see trace elements or trace minerals. Cobalt is a constituent of vitamin B12. Its deficiency leads to macrocytic anemia. Chromium, potential activator of insulin. It leads to impaired glucose tolerance in its absence. Fluoride, constituent of bone and teeth, has major role in dental caries. Iodine, thyroid hormone synthesis. Yes, thyroid enlargement and decrease in T4. Cretinism happens in case of absence of iodine. Iodine plays major role in making T3 and T4. In absence of iodine, there is no T3 or T4. Molybdenum, yes, cofactor for xanthine oxidase, which is an enzyme of uric acid formation or purine degradation, and sulfide oxidase and aldehyde oxidase. So it leads to severe neurological abnormalities and xanthinuria. Selenium, it's a cofactor of glutathione peroxidase, deiodinase, thyroidexin reductase, antioxidant along with the vitamin E. Keshan's disease is exclusive because of deficiency of selenium, cardiomyopathy, heart failure, straight and muscle degeneration. Zinc, it's a cofactor for carbonic anhydrase, carboxylpeptidase, lactate dehydrogenase, alcohol dehydrogenase, and alkaline phosphatase. So because of the absence of zinc, you will be getting growth retardation. It has major role. Zinc has major role in taste and smell. So you have decreased taste and smell, alopecia, dermatitis, diarrhea, immune dysfunction, failure to thrive, gonadal atrophy, congenital malformation, impaired wound healing also you can make out. Manganese, if you see, cofactor for arginase, carboxylase, kinase, enolase, glucosyl transferase, phosphoglucomutase, which is required for RNA polymerase. So impaired growth you can make out, skeletal development, reproduction, lipid and carbohydrate metabolism, upper body rashes you can make out. So now we'll talk about the transporters, glucose transporters. What's their major role? Glucose transporters, GLUT1. So we have already learned that so many glucose transport. What for? So different organ, they have different transporters. If you see GLUT1, there are different types of transporters which allows the uptake of glucose. GLUT1, GLUT2, GLUT3, GLUT4, GLUT5, GLUT6, GLUT7, and sodium dependent glucose transporter are also there. So the location and function is very important. GLUT1 transporter, where and all it is present: brain, kidney, colon, placenta, RBCs. So basal glucose uptake, they don't have any restrictions. Whatever the glucose is coming, they just take it. GLUT2, it is more of like a sensory type. Okay, it is exclusively present in liver and pancreas. Okay, so they will analyze the fluctuations of the glucose in the blood, and they release insulin from pancreas, and at the same time stops the mechanism of gluconeogenesis in the liver. So liver sinusoids, membrane beta cells of pancreas. Okay, they have in liver removal of excess of glucose from blood, in pancreas regulation of insulin release, low affinity and higher Km. GLUT3, mainly neurons, placenta, kidney, high affinity for glucose. GLUT4, heart, skeletal muscles and adipose tissue, insulin dependent. So one key point here to remember, that is which glucose transporter is under the influence of insulin. So insulin dependent glucose transporter is type 4. GLUT4 is the transporter which is under control of insulin. If insulin is not there, GLUT4 will not work. As our main body composition is of muscles, skeletal muscles having GLUT4 transporter in their cells, as insulin is absolute requirement. In absence of requirement, this glucose will not taken by the muscles, skeletal muscles and adipose tissue. It start producing, I mean, like, it leaves high blood glucose levels, leads to type 2 diabetes. GLUT5 present in small intestine, testis and sperm, primarily fructose transporter. It is GLUT5. GLUT6 is in a spleen and leukocyte, possibly no transporter function. GLUT7, liver endoplasmic reticulum, glucose transporter in endoplasmic reticulum. So these are the thing. And sodium dependent glucose transporter is there. It is a GLUT7 actually. This requires energy. GLUT7, otherwise known as sodium dependent glucose transporter, which requires sodium. Uh, is like co-transporter, and it is energy dependent. So far up to GLUT6, they are all like passive, and there's active glucose transporter. GLUT7, again you can get a question. Active glucose transporter, GLUT7 is a active glucose transporter which requires energy, and sodium potassium pump is used here, which requires energy. Now we'll concentrate on fat soluble vitamins. Vitamin A, which causes the active forms. Vitamin A: retinol, retinal, retinoic acid. So these are the three forms. Retinal is a alcohol form, retinal is aldehyde form, and retinoic acid is a acidic form. So retinol is required for vision. Okay, this retinal aldehyde form is required for vision, and normal reproduction and differentiation and antioxidant activity mainly by retinoic acid. Retinol alcohol form is mainly for storage and transportation. And growth and differentiation is by retinoic acid, and vision is by retinal. Aldehyde form is required for vision. How? 11-cis retinal. Remember, 11-cis retinal along with opsin. Opsin is a protein part here. Non-protein part here is 11-cis retinal. Rhodopsin is a visual pigment present in rod cells of the eye, which has got a pigment rhodopsin. Rhodopsin is a combination of protein and non-protein part. Protein part is opsin, non-protein part is 11-cis retinal. So because of the deficiency of vitamin A, what you will get? Nyctalopia, xerophthalmia, keratomalacia, dry and scaly skin, bite out spots. Yes, retinoic acid is required for maintaining healthy smooth epithelium. Because of absence of this, you will get dry and scaly skin, bite out spots, keratin debris, foamy appearance in the conjunctiva. So this is the one you see here. White triangular colored plaques on the cornea of the eye or conjunctiva of the eye is a beginning of the vitamin A deficiency, leads to keratomalacia. Conjunctiva become dry and swollen. Finally, you may lose the eyesight. Vitamin D, if you're talking, there are three forms: calciferol, ergocalciferol, and cholecalciferol. Calcidiol, calcitriol. Cholecalciferol, which you will be getting vitamin D3 from the skin, and the skin has to transport to the liver to get activate to 25 hydroxylase enzyme deficiency, and then 1 alpha hydroxylase deficiency, 1 alpha hydroxylase is present in the kidney, so make it to active. So 1 alpha hydroxylase is required, 25 hydroxylase is also required for making active vitamin D. Calcidiol, in general, cholecalciferol is having one hydroxyl group in the third carbon. In liver and in kidney, there is addition of two more hydroxyl group is in the 25th carbon and the first carbon. So total three hydroxyl group is there. So active form is calcitriol, which is having three hydroxyl group. Calcidiol is having only two hydroxyl, which is not active. So intestinal absorption of what for vitamin D we require. Vitamin D is mainly for acting at the intestine level, acting at the bone level, acting at the kidney level. So three organs it lacks. One is intestine, second is bone, and kidney. What is the activity? It synthesizes call binding proteins in the intestine. So what it will do? It increases calcium binding proteins in the intestine so that more calcium uptake. Okay, it increases calcium absorption from the intestine. And second thing, bone mineralization. It will allow calcium to deposit in the bones to make it stronger. Okay, and third thing is calcium is useful product to us, so it will ask kidney not to excrete it, so it will increases calcium reabsorption. So at three different organs it will work. Vitamin D acting on intestine, bone. If you see, there is a easy breakage of bones for small hit also. Vitamin A, the chemical name is tocopherol. Okay, and it is again an antioxidant. Neurological disorders such as hemolytic anemia you can make out in absence of vitamin E. Vitamin K, there are again three types are there: vitamin K1, K2, K3. Vitamin K1 is phylloquinone, which is present in plants, and vitamin K2 is a menaquinone, which is synthesized by intestinal bacteria. Okay, this is the only fat soluble vitamin. Remember, only fat soluble vitamin synthesized by intestinal bacteria, that is menaquinone. A menadione is a commercial form. It's synthesized in industries. Okay, it's a commercialized form produced in the labs. And what is the thing? The main function of vitamin K is it is activated form, acts as cofactor for the gamma carboxylation of glutamate. So that means here we have already learned that clotting factors. Blood clotting factors are the proteins which will be synthesized in inactive forms. So for their activation, they need post-translational modifications. One such post-translational modification helping agent, that is vitamin K, gamma carboxylation, prothrombin to thrombin. Okay, so factors like 2, 7, 9, 10, protein C and S all require these gamma carboxylation, where CO2 will be added to form mesh-like structures and aids in blood clotting. So deficiency of vitamin K leads to neonatal hemorrhage with increased prothrombin time and normal bleeding time. And to talk about water soluble vitamins here, vitamin B1, it is coenzyme form, not cofactor. Thymine pyrophosphate, TPP in short. So the enzymes which are dependent on thymine pyrophosphate, that is pyruvate dehydrogenase, alpha ketoglutarate dehydrogenase, branched chain keto acid dehydrogenase. So pyruvate dehydrogenase is the enzyme of connecting link between glycolysis and TCA cycle, whereas alpha ketoglutarate dehydrogenase is the enzyme of TCA cycle, whereas branched chain keto acid dehydrogenase is the enzyme of branched chain amino acids metabolic aspect, whereas trans ketolase is the enzyme of HMP shunt. And its deficiency leads to beriberi, Wernicke-Korsakoff syndrome. Beriberi, we have different types. Dry beriberi is there, wet beriberi is there. Wet beriberi, you can make out edema. Dry beriberi is more of like neurological symptoms. Wet beriberi, we have cardiac dysfunctions. Okay, cardiomegaly, increase in systemic BP. Okay, all these features you can make out in wet beriberi. In dry beriberi, more of neurological symptoms we can make out. Another thing, mixed beriberi is there, that is Wernicke-Korsakoff syndrome. Both and pit edema you can make out in wet beriberi. Okay, and infantile beriberi is because of deficiency of thymine in mother's milk. And Wernicke-Korsakoff syndrome exclusively you can see in chronic alcoholics who consume alcohol on daily basis, which the alcohol interferes with the absorption of thiamine in the intestine. So they are prone for vitamin B1 deficiency. Vitamin B2 riboflavin, the coenzyme forms are FAD, FMN. They are also involved in all of the dehydrogenase require FAD. And the main characteristic deficient symptoms: chelosis, inflammation of lips, scaling and fissures at the corners of the mouth, magenta colored tongue. Magenta colored tongue. So when they show you the image of magenta or purple colored tongue, or chelosis or cracks at the fissures of or corners of the mouth, it is a characteristic symptom of B2 deficiency. Seborrheic dermatitis, again one of the characteristic feature of B2 deficiency. B3, the active forms are NAD, NADP. Again, dehydrogenase require NAD, NADP. So main characteristic feature is pellagra, which is characterized by dementia, diarrhea, dermatitis. So it can be represented with the dermatitis Castle's necklace. If you can make out easily here, this is a Castle necklace. So dermatitis is a prominent feature. See here, this is a dermatitis. So this is a Castle necklace. Again, one of the favorite image based question supposed to be asked and repeatedly tested also already. And vitamin B5, pantothenic acid, the coenzyme form is coenzyme A. The main deficiency characteristic symptoms: burning feet syndrome, dermatitis, alopecia. Vitamin B6, PLP, pyridoxal phosphate, transamination enzymes like ALT, AST, heme synthesis, ALA synthase, decarboxylation reactions. So that means B6 is mainly required for protein metabolism. ALT, AST, these are the liver enzymes which dependent on B6, and transamination reaction, that means protein metabolism, and heme synthesis. First, succinyl CoA and glycine combine to form ALA synthase, which dependent on PLP, decarboxylation reactions, and glycogen phosphorylase, glycogen degradation, which is also require PLP. So that is importance of B6. So here, sideroblastic anemia, you can make out convulsions, peripheral neuropathy are the characteristic features of B6 deficiency. B7, biotin. Biotin is mainly required for carboxylation reactions. The main enzymes are pyruvate carboxylase, propionyl CoA carboxylase, acetyl CoA carboxylase. Pyruvate carboxylase is enzyme of gluconeogenesis. Propionyl CoA carboxylase is a enzyme of cholesterol. Acetyl CoA carboxylase is again fatty acid synthesis enzyme. So characteristic features in case of deficiency: alopecia caused by long-term antibiotic use or excessive. Ingestion of raw egg. So the avidin. So antagonist of B7, that is avidin, which is present in raw egg. So if you want to get the benefits of B7 from the egg, you should boil the egg before consuming. Okay. Vitamin B12, cobalamin, homocysteine methyltransferase, methylmalonyl mutase. These are the two key enzymes in our body to utilize B12 for enzyme functions. Megaloblastic anemia is a correct feature. Peripheral neuropathy, serum homocysteine and levels will be elevated. Methylmalonic acidemia is also you can see in vitamin B12 deficiency. And vitamin B9, tetrahydrofolate, it is mainly for synthesis of DNA and RNA. Megaloblastic anemia, because B9 is not there, B12 is not there, obviously it leads to B9 deficiency because of folate trap. Neural tube defects you can make out, and increase serum homocysteine levels as folate trap is not working. Vitamin C, again it is an antioxidant. Iron absorption, yes. If you want to absorb iron, it's supposed to be in ferric form, but ferrous form, but the dietary iron is in ferric form, Fe+3 form. So vitamin C in the diet converts ferric form of iron to ferrous form and make it available for the absorption. And post-translation modification of collagen, yes, maturation reactions for maturation of collagen, hydroxylation of proline and lysine to form hydroxyproline and lysine. So because of deficiency of vitamin C, you have scurvy. Image again image based question, target for image based question. Swollen gums, poor wound healing, easy bruising, hamarthritis, corkscrew hair. So that's all about vitamins. And if you are talking much about nucleosomes, electron transport chain inhibitors. So if you see here, complex 1 inhibitor, it is piercidin, amobarbital, and rotenone. Rotenone is a fish poison. Okay, and complex 2 inhibitor, that is malonate, complex 3 inhibitor, that is British anti-leucite or antimycin A, and complex 4 inhibitors, hydrogen sulfide, carbon monoxide in uh, cyanide, and complex 5 inhibitor, that is oligomycin, which inhibits ATP pump. So enzymes inhibition, if you make out, so competitive inhibition, uncompetitive inhibition, and non-competitive inhibition. So Km values and Vmax values supposed to be. So in case of competitive inhibition, the again favorite topic for the questions. So Vmax is same in competitive inhibition, Km increase. If you see here, this is a normal one. In case of competitive inhibitor, so Km will be increased, okay, and Vmax will be same. If the same, in case Vmax decreases in uncompetitive inhibition, Vmax decreases, Km will be same, okay, Km will be the same, and Vmax will be decreases in case of non-competitive inhibition. In competitive uncompetitive inhibition, Vmax and Km will be remains same. So that is the thing. So what to remember, in competitive inhibition, Vmax same, Km increases. In non-competitive, Km will be the same and Vmax will be decreases. In uncompetitive, both remains same. And isoenzyme forms, if you're talking about LDH1, H4 type is present in heart muscles. In case of cardiac infarctions or myocardial infarctions, which type of which fraction of LDH will be, is LDH4 will be elevated. And for liver enzyme, ALT, AST will be, ALT especially is a liver marker and ALP is a bone marker. In obstructive jaundice, ALP levels will be more. And another thing regarding molecular biology, the question tRNAs, it is a clover leaf like model. It has got 4 arms. One is acceptor arm, which has got CCA, where amino acid will be attached, and DHU arm, and anticodon arm, where mRNA codons will be anticodons. So mRNA, so this is anticodon. The codons are present in the mRNA. So according to that, anticodons are there in the tRNA. That is, this is anticodon arm, and there is extra arm and pseudoiridine arm, which has got T-Cyc arm. So modified form of bases will be there in T-Cyc arm. So that's all about last minute revision or one-liners from almost all the topics we have covered, and I'll be coming up with more of the case-based questions in YouTube promotions. You can follow it up. So according to me, I followed it. I have covered entire biochemistry at a glimpse. Hope it is fruitful for your preparation. If any query, anything, please drop your message in the chat box so that I can try to explain in better way. And if you want still contact me, you can get me in touch with the number. My WhatsApp number is 7095869742. This is my number. You can anytime drop me a message so that I can give you a voice message explanation, otherwise any image based explanation. So that's all for today's session. Thanks.